This is the final screencast on the nervous system content. In this screencast, we're going to finish off by talking about the neuromuscular junction. As I said previously, the neuromuscular junction obviously has aspects of neuronal and muscular control. We'll focus on neuronal for this screencast, and in the next set of screencasts, we'll talk about the corresponding muscle side of the neuromuscular junction. This again probably signposts the next set of lectures when we talk about muscle physiology. There are basically three types of muscle: skeletal muscle, smooth muscle, and cardiac muscle. And what we're going to do here is just focus on the neuronal input into each one of these types of muscle. So skeletal muscle differs from cardiac and smooth muscle in the fact that it's neurogenic. So what this means is that the origin of control is neuronal rather than myogenic, where the control is instigated in the muscle system. So skeletal muscle contraction is neurogenic. Each muscle fibers receives a single excitatory alpha motor neuron. So we've talked about motor neurons before. I'll show you the different types of motor neuron fibers. Skeletal muscle, the neuromuscular junction, receives an alpha motor neuron input. Forms one neuromuscular junction per muscle fiber, and this is called a motor unit. The vast majority, around about 98% of fibers, form this motor unit where you've got one neuromuscular junction per muscle fiber. Again, we'll talk about this a little bit more when we consider muscle physiology. Importantly, again, for neuronal control, acetylcholine is a transmitter, and postsynaptic nicotinic acetylcholine receptors mediate the response. So by contrast, smooth muscle and cardiac muscle contraction is said to be myogenic. So it originates with the muscle and it does not rely on neural input to display intrinsic rhythmical contraction. Again, maybe as I said here, smooth muscle also often receives innervation from the sympathetic and the parasympathetic nervous system, parts of the autonomic nervous system, which we talked about earlier. So smooth muscle, maybe around things like the bronchioles, has an important aspect of autonomic nervous system control. Cardiac muscle contraction is also myogenic, but different parts of the heart have different inputs. And again, we'll talk about this a little bit in our in the muscle physiology lectures and some other lectures that you'll get as part of PM1A as well, I think. There's something called a pacemaker region. Again, we'll talk about this in a little bit more detail. You know that the heart's got a pacemaker, and that also receives an important innervation from the sympathetic and parasympathetic nervous system. This is maybe a table that I'll introduce here and show you in maybe other parts of the course as well. What it does is it tells us a little bit about the difference between the three types of muscle: skeletal, cardiac, and smooth. The reason that smooth muscle is called smooth is because it doesn't have these stripes or striations that you see in skeletal and cardiac muscle. The speed of contraction of skeletal muscle is very fast because it's under this neurogenic control, whereas cardiac and smooth muscle is slower. Again, importantly, the neurogenic aspect of skeletal muscle allows it to be under voluntary control. So these goal-directed movements that we talked about already are due to the fact that skeletal muscle control is voluntary. If I want to move my hands around, if I want to vocalize, I'm thinking about it, I'm doing it voluntarily. Whereas cardiac muscle, hopefully controlling my heartbeat, I'm not thinking about that. Smooth muscle, maybe around my bronchioles, breathing, around my gut, I'm not thinking about controlling that. It's under involuntary control by the autonomic nervous system, as we mentioned, whereas skeletal muscle is controlled by these alpha motor neurons and these A alpha fibers in particular. If you want to know a little bit more about some of these fibers, A alpha are involved in muscle movement, proprioception that we talked about, and they tend to be quite wide diameter because they're highly myelinated. Again, this is probably something that we talked about when we talked about myelination when we talked about the action potential. So A alpha fibers have the widest diameters because they've got the most myelination, through to A beta fibers, which are involved in mechanoreception, A delta fibers, which are involved prominently in pain, as are C fibers, and B fibers are involved in the autonomic nervous system. Really, the take-home message for this lecture is that neurons have different amounts of innovation, or different nerves innervate different types of muscle and other parts of the physiological systems that you're going to hear about in PM1A. Myelination determines not only the diameter, the thickness of each fiber, but also the speed in which it conducts, the conduction velocity. Yeah, and that's really just to highlight from that previous table that we're talking about A alpha fibers when we're talking about neuronal innervation of the neuromuscular junction. Again, maybe a slide that I'll pick up in the lectures on muscle physiology, but what we want to do here is we want to focus on the neural input. So as we've said several places before, nerve fibers coming out of the central nervous system from these ventral horns through the ventral root are called efferent fibers, and they innervate individual muscle fibers at the neuromuscular junction. More detail to follow when we look at the in muscle physiology, as I said. What we probably want to do here is just maybe focus down on these motor end plates, and what we can see is these nerve terminals innervating individual muscle fibers. So this is a neuromuscular junction, sometimes called the motor end plate. I've probably used that phrase as well. Some textbooks call it the motor end plate because we're dealing with the motor nervous system. So yeah, this is an important concept maybe to grasp. A single motor unit may have between 3 and 1,500 muscle fibers. So you can see there are a number of muscle fibers here, but individual fibers receive one motor neuron input. So when we draw a diagram of an individual, when we draw a diagram of a neuromuscular junction, we should be showing an individual fiber going into or receiving one motor neuron input. And that's what's shown here. So if we think this is a single fiber and this is going into this single input here. We'll have a look at this, as I said, in detail in muscle physiology lectures, but let's focus on the neuronal side of it. And hopefully we're going to see that this pre-synapse is very much similar to the diagrams that I've shown you before. And we'll put some labels on it, hopefully, so that you can equate this motor neuron presynaptic terminal with the presynaptic diagrams that I gave you before. So we can see it contains vesicles, in this case containing acetylcholine. Acetylcholine is released across the synaptic cleft, around about 20 nanometers wide, as I showed you before, and acts on postsynaptic receptors, in this case on the motor end plate. And remember, those postsynaptic receptors are nicotinic acetylcholine receptors. So a few other terms maybe to, again, we'll pick up these in the muscle physiology because they're terms about the muscle rather than the nerve, but the plasma membrane of a muscle cell is called a sarcolemma instead of a plasma lemma because sarco is to do with muscle. This is the motor end plate. These are called T tubules. So if you look at this shape here, it looks like a T shape. A T tubule is basically the invagination that separates one motor end plate, again maybe from other motor end plates on either side here. And these are called terminal cisternae. Cisternae are just these stores, if you like, here, which contain calcium. And you can also see some idea of some filaments. We'll tell you in maybe next week's next set of screencasts that these are actin and myosin filaments, and that they slide over each other in order to cause muscle contraction. This distance between, this is called a Z line because it's Z-shaped, this red Z line, and this next one is called a sarcomere. So again, sarcro is a term that's to do with muscle. Yeah, here's just actin and myosin labelled. Myosin shown here in red. The thin blue filament actin shown here, and yeah, there's a little diagram, a little label showing you that's called a Z line, as I've said already. So let's have a look at the sequence of events. As I said, you know, in terms of the neuronal input into the muscle. So action potentials from these alpha motor neurons depolarize the terminal. Again, as we've said before, depolarization opens voltage gated or voltage dependent calcium channels. Calcium comes down its concentration gradient, and we get calcium dependent protein-protein interactions which trigger vesicle fusion. Acetylcholine is released. This process is called exocytosis. So exocytosis is just the release of a transmitter substance at the presynaptic terminal, a term that we've probably used before. Acetylcholine diffuses across this narrow synaptic cleft, principally activates nicotinic acetylcholine receptors, certainly at the neuromuscular junction, opens the receptor ionophore. Remember, we showed you a diagram of the nicotinic acetylcholine receptor that had a gate that opened and shut when acetylcholine binds. Sodium comes down its concentration gradient, depolarizes because positive charge is coming into the cell, so depolarizes the membrane. So again, maybe this is another term which we need to make sure that you understand. We talked about action potentials, we talked about excitatory postsynaptic potentials and inhibitory postsynaptic potentials. This is another type of potential called an end plate potential, an EPP. And essentially what you can think about is the EPP is analogous to the EPSP. The EPSP occurs at neurons. The EPP occurs at muscles. So exactly the same. In this case, we've got a nerve communicating with a muscle instead of a nerve communicating with another nerve. Nerve to nerve would be an EPSP. Nerve to muscle is an EPP, end plate potential. So, you know, just make sure you recognize these terms and that they're different to an action potential. That's a good thing to remember. EPPs summate like a EPSP, and if a threshold is attained, trigger an action potential. So absolutely analogous to what the situation we talked about when we talked about synaptic integration for EPSPs. This is the rest of the sequence of events. These are the ones that we're going to cover in the muscle physiology lecture. That's why they're grayed out. But essentially, just for completeness, I'll just go through these very quickly. So we'll tell you about, in the next set of screencasts, excitation, contraction, coupling, where the action potential, the muscle action potential in this case, instead of a nerve action potential, spreads along the sarcolemma to the T-tubules, releases calcium from the internal stores, and triggers this link between excitation, which is to do with nerves, and contraction, which is to do with muscle. So EC coupling, excitation, contraction coupling. This is the neuronal part. This is the muscular part that we'll talk about next set of screencasts in a bit more detail. We'll talk about the calcium binding protein. So there's one called troponin, which is an important calcium binding protein present on these actin fibers, the thin fibers. That calcium dependent process triggers actin and myosin fibers to interact in something called the sliding filament model. Requires energy, ATP. Maybe again, this is something that's important, and then to pick up that we've shown you on previous slide. In order to, it's very important that we break down acetylcholine, or else we get problems that can actually lead to death. Acetylcholine is continuously degraded into its component parts, acetate and choline, by an enzyme, acetylcholine esterase at the neuromuscular junction. Once acetylcholine is broken down by this important enzyme, the levels of intracellular calcium within the muscle, the sarcoplasmic calcium concentrations fall. Actin and myosin interactions are inhibited, so this sliding filament model is broken and the muscle relaxes. Okay, just a couple of slides to finish off. There are a couple of agents that we'll talk about probably in other parts of the course. Neuromuscular junctions can be blocked by poisons that cause paralysis. So paralysis when you can't move your muscles properly. There's one called curare, which is an arrow poison. We know the reason that it's important is because we derive some medically important drugs. Tubocurare is a compound that's based on our knowledge of curare. We use that in surgery. And we use it in general anesthesia and surgery, as I've said. So curare is important. We knowing a little bit about neuromuscular junction and knowing something a little bit about pharmacology, we can have these agents, which are important agents. Botox, which we'll talk about again, probably in other parts of the course. The reason that Botox is useful cosmetically, but also, of course, therapeutically, is because Botox actually prevents this fusion of the vesicle with the presynaptic membrane, and so you can effectively block a neuromuscular junction. Cosmetically, that might cause relaxation to get rid of frown lines and wrinkles and things like that. Let's say more importantly, medically, it can treat muscle spasms. Things to find out about in other parts of the course. So this is the final slide. Again, I'll just talk you through this and then we'll finish. So alpha motor neurons are important, and they, together with the muscle fibers that they innervate, that they feed into, they form a motor unit. Action potential from an alpha motor neuron causes release or exocytosis of the transmitter, acetylcholine. Acetylcholine binds to our postsynaptic nicotinic acetylcholine receptors, and in this case, the muscle generates an end plate potential. As in neurons, if the end plate potentials add up, summate, and go above the threshold, in this case, a muscle action potential fires. Muscle action potential promotes calcium release from internal stores, these terminal cisternae, and calcium release couples excitation to contraction. Good. Okay, so that's the last of the three screencasts, I think 7, 8, and 9, that we want to cover. Remember to have a look at the activities that are associated with each of the sets of screencasts. Okay, we'll end there.